But in the 1920s, a biochemist named P.A. Levine worked out its basic structure. Levine found that DNA is made up of three main components: a phosphate group, a 5-carbon sugar known as pentose sugar, and nitrogen-containing bases. These bases are of two types, purines and pyrimidines. Purines, adenine A and guanine G, pyrimidines, thymine T and cytosine C. And in case of RNA, instead of thymine, we find uracil U. Levine also discovered that these components form repeating units called nucleotides. Now, let's see how a nucleotide is arranged. The nitrogen base attaches to the carbon number 1 of the sugar, the phosphate group attaches to the carbon number 5 of the sugar, and there is also a hydroxyl group OH at carbon number 3.